Uh, MUC1 is a heavily glycosylated protein that is found, that is overexpressed on the surface of many cancers of epithelial origins. And one of the beauties of this MUC1 protein is that it has tandem repeats, 20 amino acids in the later part of my talk. Now, these are the two cartoons representing normal MUC1 and MUC1 on cancer cells. As you can see, the normal MUC1, it is quite uniformly glycosylated. But what happens in MUC1 on cancer cells is that this glycosylation are generally aberrant, shorter, and truncated. And the carbohydrate epitopes, those are found on the surface of tumor-associated MUC1, are known as tumor-associated carbohydrate antigens, or TACAs. And a few examples of the TACAs are the TN antigen, the TF antigen, and the sialyl-TN antigen. Now, the question is, can we use these TACAs as vaccines by themselves? The answer is no, because the T cell help is required for immune response, and the TACAs are not able to give that T cell help for that immune response. So therefore, to obtain the give the T cell help or to activate the helper T epitope, we need helper T epitopes on the vaccine design. Now, a few earlier strategies to get the helper T epitope was to conjugate the TACAs to large carrier proteins like KLH. Now, one of the disadvantages, there were many disadvantages. Now, one of the major disadvantages of this strategy was that the immune response, which was specific to the carrier protein, suppressed the immune response for the carbohydrate antigen. And therefore, the vaccines were not successful. Now, in one of the pioneering strategies by Professor Grijan Boons, who happens to be my previous advisor, he did this work at the Complex Carbohydrate Research Center. What he did was he formulated a 3-component vaccine. The 3-component vaccine consists of the carbohydrate antigen, cancer antigen, TN, a helper T epitope, YAF, which is found on the outer membrane protein of Neisseria meningitis, and that activates the helper T cells, and an immunoadjuvant, the PAM3 cis. Now, this 3-component vaccine was able to break the self-tolerance of the TACAs and activate the helper T cells, and therefore, the immune response observed for this 3-component vaccine was robust. So that's one way how you can make a vaccine more immunoresponsive. Now, let's switch gears and see there is another way of making a vaccine more potent, and that is by using xenoantigens. Now, xenoantigens are antigens to which the human body contains naturally occurring antibodies. One of the most highly studied xenoantigen is alpha-gal. Now, alpha-gal epitopes have been conjugated to many vaccines, like the vaccines for HIV, vaccines for flu, and how it works, I'll show you in the next slide. So what happens when you coat a vaccine? So this is an example of a flu virus vaccine. When you encapsulate a vaccine with the alpha-gal epitope, there are naturally occurring alpha-gal antibodies in the serum. What happens is that the naturally occurring alpha-gal antibodies bind to the alpha-gal epitopes on the vaccine. The Fc portion of this antigen-antibody complex is recognized by the Fc gamma receptors on the antigen-presenting cells. Thereby, the entire vaccine is internalized, better processed, and displayed on the major histocompatibility class 1 and class 2 molecules towards the T cells, and therefore we get a very high immune response against the vaccine. So this mechanism is known as antibody-dependent antigen uptake mechanism. Now, in another pioneering study by Professor Jeff Gildersleeve at NIH, he did glycan arrays for many different glycans. And what he found out was that actually the human serum contains the maximum amount of antibodies against rhamnose than alpha-gal. And therefore, what we did was that we bounced on this idea, and we thought that can we put these concepts together? Can we have a helper T epitope YAF that was designed by Professor Boons, and can we conjugate the cancer antigen to the rhamnose epitope? So we would design a 3-component vaccine which contains the cancer antigen Tn, the helper T epitope YAF, and the rhamnose epitope. So the idea behind this vaccine was that there will be naturally occurring anti-rhamnose antibodies in the serum that would bind to the rhamnose epitopes. The Fc portion of this antigen-antibody complex will be recognized by the Fc gamma receptors on the antigen-presenting cells. Thereby, the entire vaccine would be better internalized on the antigen-presenting cells, better processed, displayed on the MHC class II molecules, and we will get a better immune response. So that was our idea. And we actually synthesized 2 different molecules. One molecule, which is a 3-component vaccine, which contains the Tn antigen, can I get another?
You'll use that one. I think that's on top.
All right, thank you. So we designed another rhamnose-containing vaccine which contains the Tn antigen, the YAF-Tn helper T epitope, and the rhamnose epitope. And we designed another control vaccine which contains only the Tn antigen and the YAF epitope. And these were the outcomes that this vaccine, the rhamnose vaccines, was able to give better antibody response and higher amounts of anti-Tn antibodies to this particular vaccine. So this vaccine was successful at generating antibodies, and also it was successful in generating a T cell response. So therefore, we were successful in generating this 3-component vaccine with the rhamnose. So I'm not going to talk about this particular vaccine. It was published in Journal of American Chemical Society a couple of years back. Now, when we had this success in this 3-component vaccine, we thought, can we make this vaccine better? And how can we make this vaccine better? So we wanted to introduce multivalency in the vaccine design. So why? Because, and how we would introduce multivalency is that we thought we could either have multiple Tn epitopes and multiple rhamnose epitopes on the same construct, or we could formulate the vaccine into liposomes. And the liposomes will give us the desired multivalency in the vaccine design. And also, there was another aspect we wanted to introduce in the vaccine design. Previously, we were using just the carbohydrate antigen Tn, but right now we wanted to use the entire MUC1, glycosylated MUC1 with the Tn as the helper T epitope. And we had literature precedence because recent discovery by Professor Finn, she reported that the immune response with this glycosylated MUC1 variable number tandem repeat was higher than the immune response when just this MUC1 variable number tandem repeat was used. And therefore, we hypothesized that we wanted to synthesize a multivalent vaccine which would contain the MUC1 variable number tandem repeat conjugated with the TACA Tn instead of the YAF Tn which we used in our previous vaccine. So this is more homologous to an actual cancer scenario than this heterologous YAF helper T epitope. So that was our hypothesis. So this was our liposome design. So we would design the liposome in such a fashion that we will have the MUC1 variable number tandem repeat conjugate with the TACA, and we would conjugate it to the PAM3 cis, which is an immunoadjuvant which stimulates the TLR2. So that's an adjuvant built in the vaccine design itself. Now, we would introduce the rhamnose epitopes on the vaccine by conjugating it with the lipid cholesterol. And this would give us multiple copies of the rhamnose and the MUC1 Tn on the liposome. And the idea behind this is again the similar that the anti-rhamnose antibodies would bind to the rhamnose epitopes. The Fc portion of this antigen-antibody complex would be recognized by the Fc gamma receptors on the antigen-presenting cells. The entire liposome would be better internalized, processed, and we would get much higher antibody response to this MUC1 glycopeptide. So that was our hypothesis. So the first part of this project was the synthesis of the components, individual components. So the first part of the synthesis was the L-rhamnose cholesterol conjugate. We started with cholesterol, did a tosylation reaction, and then we conjugated it with the tetraethylene glycol to give the tetraethylene glycol cholesterol. Then we had the per-acetylated rhamnose molecule, and we did a glycosylation with BF3 etherate, and we formulated the acetylated version of the rhamnose cholesterol conjugate. Then we did Zempling condition deacetylation with sodium methoxide and methanol, and we got the cholesterol with the tetraethylene glycol rhamnose conjugate. So that's one part of the liposome. The next part was the synthesis of the alkyne functionalized PAM3 cis. So we started with the cysteine dimer, which is protected with the tetrabutyl. We first did an FMOC protection. Then with the zinc and methanol in sulfuric acid, what we did was cleaved off this disulfide linkage, and we coupled it with the S- glycidol to give this particular molecule. Then the free hydroxyl, 2-hydroxyl groups were palmitoylated with palmitic acid in DIC and DMAP to give the PAM2 cis, which was protected by the tertiary butyl group at the C terminal and the FMOC group at the N terminal. Then we sequentially deprotected the tertiary butyl group by TFA and coupled it with the propargyl amine to give the alkyne handle at this end. And then we preferentially cleaved off this FMOC group on the nitrogen and did another palmitylation reaction with palmitic acid, PIBOP, and HOBT to put the another palmitic acid group here to give the final molecule as PAM3 cis with an alkyne handle. So that's one part of the molecule. Now we wanted to make this PAM3 cis Mach1 variable number tandem repeat TACA conjugate, and how we did it, we followed a solid phase peptide synthesis by using FMOC chemistry. We sequentially added each amino acid and then added the TACA. And then with the resin cleavage here, and we had a terminal azide group. Why we had this terminal azide group? Because we would have another alkyne functionalized PAM3 cis, and we will do a click reaction to conjugate these two molecules together. So we had this alkyne functionalized, azide functionalized PAM Mach1 variable number tandem repeat TACA. And then we did a click conjugation with copper sulfate, sodium ascorbate, and TBTA to get the final another component of our vaccine, which contains the PAM3 cis and the Mach1 with the TACA. So that's another component of the liposome. Now, after we had both the components, we wanted to do a liposome preparation. So how we do it? This is straightforward liposome preparation. What we do is we make lipid stock solution in chloroform and mix them in glass vials, and then the chloroform layers are evaporated to get thin lipid films. Now we hydrate it with Heaps buffer, and then we agitate it at 43 degrees centigrade for 40 minutes, and then subject to freeze-thaw cycles, and then extrude 21 times through this instrument, which is known as a LipoFast Basic, and which is fitted with a 100 nanometer polycarbonate membrane. So once we have the liposomes, we wanted to characterize whether we have the liposomes exactly similar as we want it to be. So we wanted to study the stability and homogeneity and size characterization. For that, we used dynamic light scattering experiments. And then we wanted to do the scanning electron microscopy imaging of those liposomes. And we have the rhamnose epitopes and the Mach1 epitopes on the surface of the liposomes. But how do we know that exactly those epitopes are there? Then we did an antibody binding experiment to prove that. So here are the results. So this is the dynamic light scattering measurements. This is just the buffer, and this is for the liposomes. So we see that it is around 100 nanometer in diameter. And then these are the scanning electron microscope imaging of the liposomes, and we see that the size of the liposomes is around 100 nanometers. So these liposomes were stable for 2 degrees at 4 degrees centigrades. Now we did an antibody binding experiment. So what we did was that we took the liposomes and then we incubated them with just normal mouse serum. And then we incubated them with anti-mouse antibodies, which is FITC conjugated. So these doesn't bind to anything. But these this is the same liposome which contains the rhamnose epitopes. We incubated them with the serum which contains anti-rhamnose antibodies. And then we incubated them with a secondary antibody which recognizes the mouse antibody, which recognizes the mouse antibody and which is also FITC conjugated. And we see that these antibodies bind to it. So therefore, the rhamnose epitopes are on the surface of the liposomes. We again took the liposomes and we incubated with anti-MUC1 antibody and then used a secondary antibody which contains the FITC. And we again see that the surface of liposomes contains the MUC1 epitope. So therefore, the surface of liposomes contains the rhamnose epitopes as well as the surface of the liposomes contain the MUC1 epitopes as well. So therefore, the liposomes are exactly similar to what we wanted them to be. Now, once we know that, then we did the immunization study. We did the immunization study in mouse. We had 4 different groups of mouse. Now since we want to test the rhamnose vaccines to it, we have to make sure that the mouse are producing anti-rhamnose antibodies. Now, in our previous study, in one of our previous studies, we showed that normal mouse, which are laboratory mouse, they do not by itself make those anti-rhamnose antibodies. So therefore, we have to introduce artificially by immunization the anti-rhamnose antibodies to mimic the human beings. So we had 4 different groups of mice, and the first step of the vaccination study was the immunization with the rhamnose ficol. So group A1 and B1, they were non-immunized, and groups A2 and B2, they were immunized with the rhamnose ficol. And after like 4 immunizations, I'll show you the results. We showed that the mouse right now contains anti-rhamnose antibodies and they are ready for the vaccine challenge. Now groups A1 and A2 were challenged with the PAM36 MUC1 TN liposomes, whereas groups B1 and B2 were challenged with the PAM36 MUC1 TN plus the rhamnose liposomes. So here are the results. So this is the anti-rhamnose antibody titers after 4th boost with the rhamnose ficol. We see that the groups of mouse, the A2 and B2, which were immunized with the rhamnose ficol, they showed a 25-fold greater titers of anti-rhamnose antibodies than the groups A1 and B1, which are non-immunized. So now the groups A2 and B2 are producing the anti-rhamnose antibodies for the vaccine challenge. Now, after the vaccine challenge, what we showed was the groups B2, which was previously immunized with the rhamnose ficol and later challenged with the liposomes, with the MUC1 and the rhamnose, that showed a 8-fold increase in the amount of anti-MUC1 TN antibody titers than the other groups, which were either not immunized with the rhamnose ficol, or immunized with the rhamnose ficol, but not challenged with the vaccines containing the rhamnose. So therefore, having the rhamnose as well as the MUC1, having the rhamnose on the vaccine in the mouse where the mouse are producing anti-rhamnose antibodies, it is actually producing higher amounts of anti-MUC1 TN antibody titers by the antibody-dependent antigen uptake mechanism. All right, sure. Thank you so much. So again, for the anti-TN antibody titers, we see the same thing, that the groups B2 that also was immunized with the rhamnose ficol and later challenged with the rhamnose vaccine, that also showed higher amounts of anti-TN antibodies. Now we did a competitive binding experiments where we incubated the serum with some free TN before doing the ELISA. What we found out that the anti-MUC1 antibodies in the serum are now competing with the antigen on the plates as well as the free TN antibodies. So therefore, when we do not have any free MUC1, we see an absorbance here. When we add free TN to a concentration of 10 to the power minus 5, absorbance decreases. When we again go to 10 to the power minus 4, again absorbance decreases, and when we go to minus 3, again absorbance decreases. So therefore, the antibodies are competing against the free MUC1 as well as the MUC1 on the ELISA plates. So there is another experiment where what we did was we took an actual human leukemia U266 cells and we incubated with anti-MUC1 antibody, and what we saw that we see a shift in the cells, in the population of the cells. Now, here in this study, what we did, we used the same cells and we incubated them with just non-immunized mice sera. We see the population of the cells here. But when we use the sera from the mouse, which from the groups B2, we see that the population of the cells shift. So therefore, the antibodies generated after vaccination are recognizing the MUC1 on the leukemia cells. So for the conclusions, we were successful in preparing the vaccines. And then the vaccines, we will characterize the vaccines so that we know that it contains the same MUC1 as well as the rhamnose epitopes on them. And there were higher amount of anti-MUC1 and anti-TN antibodies for the groups of mice which were immunized with rhamnose ficol and later challenged with the rhamnose vaccine. And also the generation of the glycosylated anti-MUC1 and anti-TN antibodies and the antisera bound to human leukemia U266 cells. With that, I would like to thank my advisor, Dr. Steven Suchak, the University of Toledo, Ohio Cancer Research Associates, Elsa Pardee Foundation, and the NIH for funding. And I'd like to thank you all for giving me an opportunity here, and I would be happy to answer any questions. Thank you so much.
Okay. Thank you very much. Let me ask a question first.
Sure.
So, do you know what type of antibodies you guys got with the IgGs? IgMs?
Yes, we did antibody
subtyping.
That's isotype isolation, and what we saw that we got IgG1 as well as IgG3.
Three. So you
Yes,
got 3. Okay.
I get 3. We get 3 as well as we get IgG1 when we have probably immunized with rhamnose ovalbumin first, but with the rhamnose ficol and later challenge we got major amount of IgG3. So we did an isotype.
Do you know what the highest titer you had was about approximately? I mean, against your particular epitope, you know, I just don't, just curious, you know, because there's lots of ways to immunize with TN, and so.
Yeah, so I don't exactly remember the titers exactly because I don't have the that isotype data in this presentation,
Okay.
but there's a publication we have in bioconjugate chemistry.
Right, it's in there.
patent, so
Okay.
we have that over there. So.
Yeah, one other quick thing, you know, making these molecules, these tri-component vaccines, you know how difficult they are to
Yeah.
synthesize. I mean, how difficult are they to characterize? I mean, to actually characterize the monovalent molecules before you put them on a liposome. Did you guys do just mass spec, or do you?
We do an HPLC, and then we do mass spec.
Yes, and
then all the components, individual components, we do.
You look at that.
We look into that, like the alkyne functionalized camptresis, we could do an NMR and everything,
Yeah.
and then the peptide components, we did
Right.
HPLC mass spec, and then combine HPLC mass spec again. So
Like, what are the physical properties? Are they unusual solubilities or something? Because you have fatty, you have peptide, you have.
So
Yeah.
so this is a very important question. The most challenging reaction which I faced was getting that click conjugation of the alkyne functionalized with the peptide, where I had to use a mixture of solvents, where I used methanol, then THF, water, and I guess chloroform. So it's a mixture. So I had to try it probably a couple of times, and then I have to get it right.
Everything
so
falls off the shelf, and you.
Yeah, so because they have like very, very different solubility, but
Yeah, it's very different.
ultimately, I mean, I got it to work.
Any other questions in the back?
In the final step of the synthesis of liposome, you use click reaction.
Yeah,
The major challenge there is to remove the copper.
yeah.
So what sort of contamination do you actually have in your liposomes?
Is your question, how do I remove the copper?
How do you remove it, and what level do you end up with?
Okay,
End of
so
the day.
first, the purification, how I do it is after I run the click reaction, I run it through a LH20 column. When you run it through an LH20 column, the copper is
it's
would be separated over there. Now we do an HPLC after the fraction. So that fraction is free of copper. So that's how I separate the copper. And I know the copper is not there. And if the copper is there, that would also show up in the mass spec. But
Well, copper is known to chelate to triazole, because during the click reaction, you form triazole ring, and that is known to chelate the copper.
Yeah, that's what I know.
And because of that, it's difficult to remove.
I understand. So you're saying that the copper is going to chelate to the peptide backbone, right? I also had this particular thought in my mind before I ran this reaction. But what I believe is that the ratio, I had to formulate, I mean, I had to play around with the ratio of adding the copper and everything. So the final ratio which I had, I'm pretty much sure that since I got an accurate mass spec of my product final, I'm pretty much sure that the one which I used for making the liposomes, that doesn't contain the copper. Otherwise it would show up. But I ran the LH20 column, and then I ran the HPLC on it. And I
was going to suggest use copper-free.
No. Well,
all right.
one more.
Very nice study. The question here is with regard to the specificity of your anti-MUC1 antibodies that you generated. You mentioned in your first slides that in cancer cells, the MUC1 is decylidated, or there's a truncation of the MUC1 glycosylation. So the antibodies you generated, what's the specificity? How's it going to react? What is it reacting to?
Well, this is the one where I have this sort of slide where we have human leukemia U266 cells, right? And this is actually cancer cells. So therefore, the MUC1 which would be found here, that would be specific towards the MUC1 which is for a cancer cell. Now, when we generate the antibodies on this kind of mice, that is actually binding to that MUC1 on these cells. So therefore, I'm pretty much sure that the antibodies which are generated in mouse, those are specific towards the MUC1 common cancer cells. And this one is just normal serum. So that doesn't contain these antibodies. So therefore, we are seeing this shift.
So the specificity I'm asking is it at the full glycosylation of the MUC1, or is it truncated?
Yeah.
Well,
You'd have,
That's
to
the
have,
question.
you'd have to have all those synthetic molecules that are in it, or something
hasn't
else
done
to
it.
be able to do it. Yeah, so it'd be pretty difficult. But I mean, it does cross-react with the peptide, right?
yes.
Yeah, so, with the free peptide. Yeah.
Thank you.
Okay, I think we're going to move on. Thank you very much, Sourav. Appreciate it.